Welcome to VetReview. In this video, I'm going to talk about anthrax. Anthrax is caused by a gram-positive bacteria called Bacillus anthracis. The general features of members belonging to the genus Bacillus are they are large gram-positive rods that are capable of forming endospores. They are aerobes or facultative anaerobes that grow on non-enriched media. Most species are motile, catalase positive and oxidase negative, and they have the ability to grow aerobically and to produce catalase. Okay, so the main important points to remember are they are gram-positive rods capable of forming endospores. They are aerobes, motile, catalase positive and oxidase negative. And to distinguish between Bacillus and Clostridium, you have to look at the ability to grow aerobically and catalase positive. Okay, then let us come to epidemiology. So anthrax can virtually affect all mammalian species, including humans. So it is a zoonotic disease. Birds are very resistant to anthrax. The susceptibility is highest in ruminants, then pigs and horses, and then carnivores. So the disease is per acute in case of ruminants, acute to subacute in case of pigs and horses, and subacute or even longer than that in case of carnivores. Endospore formation is the most important factor in the persistence and spread of anthrax. Okay, so this is general vegetative cell. Under unfavorable conditions, it will convert into a very resistant structure called endospore. Okay, and these endospore can persist or survive in the soil for many decades. Okay, herbivores contact the disease by ingesting spore contaminated vegetation. So the source of transmission in case of herbivores is ingestion. Okay, other than that, cutaneous wounds or inhalation can also transmit the disease. In case of carnivores, ingesting the contaminating carcasses is the most common source of infection. In case of horses, biting flies can also transmit the bacterium. Okay, so biting flies is common source of transmission in case of horses. Then pathogenesis. So in pathogenesis, we have to look at the virulence factors. So there are two important virulence factors without which the bacterium cannot cause the disease. So first of these two virulence factors is Anti-Phagocytic Polypeptide capsule. So the bacteria is capsulated and the capsule is made up of poly, the glutamic acid. Okay, and it has anti-phagocytic property. So if the macrophage will try to phagocytize the bacteria, it won't be able to do so. And if it does, then the bacteria will not be digested inside the macrophage. So the bacterium can live and grow inside the macrophages and dendritic cells. Okay, then we have tripartite toxins, which is the main toxic substance. Okay, so capsule does not have any pathogenic property. The main pathogenic property of the bacterium comes from the tripartite toxin. So it is a AB type toxin. So A means active ingredient or active component, B means binding moiety. So here the protection factor acts as a binding moiety which binds to the edema factor and lethal factor. Edema factor mainly affects neutrophils where it will cause poor water homeostasis. Okay, that is why it is causing edema. And lethal factor will mainly affect the macrophages where it will stimulate the macrophages to release cytokines. Okay, other than these toxic components, B. anthracis also release two siderophores, bacillibactin and pectrovectin, which are responsible for scavenging the iron from extracellular matrix. Then we have pathology. So anthrax is mainly classified into three types. We have alimentary anthrax, cutaneous anthrax, and pulmonary anthrax. In case of alimentary anthrax, the source of the infection is by ingestion. Okay, so it can be of two types, oropharyngeal or intestinal. In oropharyngeal type, the main lesions are located or localized in the oral or esophageal region. Okay, so we see ulcers with infection of associated lymph nodes. We have swelling, dyspnea, and dysphagia. Then intestinal form is much more severe than the oropharyngeal type. Here we see ulcerative hemorrhagic ebomyositis or ulcerative hemorrhagic enteritis in case of cattle. So here you can see this is a picture of ulcerative hemorrhagic enteritis. Okay, then we have cutaneous anthrax. Cutaneous anthrax is very much common in case of horses and it is the most common form of the disease in case of humans. Here we have two lesions. First is the edematous swelling and second is carbuncles. Carbuncles is very much common in case of cattle and humans and much rarer in case of dogs. So what are carbuncles? They are discrete necrotic areas on the skin. Okay, and the source of infection in case of cutaneous anthrax is wound infection. Okay, so if there's some cut or wound on the skin, then the spores of the bacterium can enter there. They will enter into the wound and germinate into vegetative cells and will cause anthrax. Then we have pulmonary anthrax, in which the source of infection is inhalation of the fomites which are contaminated with endospores. Okay, and there from there they will enter the lungs. Okay, and then they will be phagocytized by the macrophages and dendritic cells, which will then spread the bacterium to the regional lymph nodes. Okay, so these are the three types of anthrax: alimentary, cutaneous, and pulmonary, and all three types will finally lead to septicemic anthrax, in which death occurs. Okay, so the main lesions of anthrax are splenomegaly, which is the pathognomonic lesion of anthrax. Okay, splenomegaly, or we can also say blackberry jam spleen. Then you have enlarged hemorrhagic lymph nodes, hemorrhages on the serosal surfaces, tarry colored unclotted blood oozing out of the body surfaces. This is the most important sign. So, necropsy should never be performed on an animal that is suspected to be died from anthrax. Okay, and how you will suspect the animal? The carcass will be very much bloated. There will be absence or presence of very poor rigor mortis, and you will see tarry colored unclotted blood oozing out of the body orifices. Okay, so this is the main indication, so that you can suspect that the animal is died from anthrax, and in this case you will not perform the necropsy. Okay, so this is a picture of the blackberry jam spleen, hemorrhagic enlarged lymph nodes, and here you can see a carcass suspected to be died from anthrax, and you can see that there's sawhorse appearance. Why? Because the animal is very much bloated. Okay, so diagnosis point to note here is that necropsy should not be performed. So how you will do the diagnosis of specimen collection? You will take the peripheral blood from the tail vein or ear vein of ruminants, or you can also take peritoneal fluid in case of pigs. Okay, after the collection of blood, you have to soak a cotton in 70% alcohol and apply it to the site of blood collection to minimize leakage. Why? To stop any environmental contamination, because the spores are very much resistant and they can survive in the soil for many decades. So we have to stop the environmental contamination at any stage. Then the primary identification, the primary diagnosis is by McFadden reaction. Here you will stain the blood smear with polychrome methylene blue, or if it is not present, then you can use gemsa stain, and you will observe it under the microscope. What you will see square-ended blue rods with pink capsule. Okay, so this is a picture of McFadden reaction. This is very much important from an exam point of view. Then isolation. Isolation is not routinely done, but for experimental purposes, you can do the isolation. Here, what you will see, ground glass appearance and Medusa head appearance. These are the two main important points. So the bacteria will grow well on sheep or ox blood agar aerobically at 37 degrees Celsius for 1 to 2 days. Okay, it will not grow on McConkey agar. The colonies are flat, gray, and granular, and this is called the ground glass appearance. Okay, this is a picture of ground glass appearance of the colony of bee anthracis. Under the low power of the microscope, the edge of the colony will appear like a Medusa head. Okay, so there's Medusa head appearance. Why? Because of curled peripheral projections at the edge of the colonies. Okay, then you have a test called Ascoli test, which is thermoprecipitation test. Again, very much asked in the exam. Here you will take 2 to 3 gram of homogenized material in a little saline, boil it and pass it through a filter paper. The filtrate is then used as an antigen in ring precipitation test. Okay, so this is a picture of Ascoli test. Then the last point is that the inverted fir tree type of liquefaction is produced by bee anthracis. So you will take a nutrient gelatin, make a stab, okay, stab the nutrient gelatin with your specimen, and after 8 days, where you incubate it aerobically, what you will see, side shoots will radiate outwards. Okay, and this is called inverted fir tree type of liquefaction. So it will give a characteristic inverted fir tree type of growth or liquefaction with side shoots radiating from the stab line. Okay, other than these tests, you can also go for animal inoculation. This is also done in case of experimental studies. Here you take a dilute sample of B anthracis of the suspected case, and you will inoculate in the tail of a rat. Okay, in case the rat dies, this will give a positive reaction. Then at last, the thing which is very much asked in the exams is vaccination. So routine vaccination of anthrax is not done, but if you have a suspected case, then the nearby cattle can be vaccinated, or if you're suspecting an animal to be exposed to anthrax, then you can also vaccinate. So suspension of living spores of uncapsulated A virulent strain of Bacillus anthracis are used for vaccination. The main thing that is asked in the exams is what is the strain, and it is Stern 34F2. Okay, so this is it for anthrax. I hope you like this video. If so, hit the like button, share this video with your friends, and subscribe to my channel. Thank you.